Hello, this is Dr. Pankaj Kumar, your mentor for the current session of NEET Bites, and we will be talking about the translocation of solutes. So far, we have just seen that in case of photosynthesis, the carbohydrate is formed, and all the carbohydrate that is being formed in the form of glucose or fructose, and eventually it is actually stored in the form of starch. But what happens that a starch cannot be translocated to the phloem? First of all, it has to get converted into some monosaccharide or disaccharide forms, then only it can be transported. Okay, so what happens that all the stored sugar is actually gets converted into transportable form, and the most important transportable form is a sucrose. So the entire translocation of solutes is going to occur through the sucrose only, right? However, there are some other forms as well. For instance, take an example of raffinose or stachiols, primarily stachiols in case of grapes. Okay, but we can say that in 90% of the cases what happens that translocation is going to occur in the form of sucrose only, right? Now, is it passive process or is it an active process? Actually, it is an active process. Okay, and the theory that is given for it is what we call pressure flow hypothesis. Earlier, you see, this hypothesis was given by Munch, but later on Munch, the very concept of Munch was actually a passive one, but now we know very well that the active loading of sugar is required, and for this energy is required. That's why we say that translocation of solute is an active process, right? So here what we understand, there are two things: one, the source, and second is the sink, right? What is source? Source is those areas in which the carbohydrates are being synthesized. Primarily it is the leaf where the photosynthetic products are going to be stored, right, or synthesized. Okay, once the synthesis happens, then this synthesized products need to be translocated to the area where it required to be consumed. So it may be, let it be say roots, where the food is going to be stored in the form of starch, or stem, or any other part where there is actual requirement. Okay, so we will just consider that how the sugar is actually being transported from source to sink, right? So you see, there are two possible ways by which sugar is translocated, right? One what we call active transport, and second what we call the polymer trap mechanism. Okay, now the sugars are actively transported into sieve elements. Of course, here energy is involved, but in some species what happens that phloem is loaded by what we call polymer trap mechanism. Okay, so here the conducting cell plasma membranes are permeable to monosaccharides and disaccharides, but not to the polysaccharides. Okay, so what happens, simple sugar merely diffuse into the conducting cells, right, and eventually they are polymerized into polysaccharides so that they cannot diffuse back out. So that is how actually it works, right? Now have a look at this diagram. So this diagram is actually talking about the source where the photosynthesis is going to happen. You can see there that this is the area where photosynthesis is happening, right, and this is the sugar is actually being converted into starch. Now what happens, cells surrounding the sieve elements, both campanule cells and other phloem parenchyma cells are important in terms of loading things. Okay, so you can see there that this is the phloem cell, and actually this is, or we can say this is the sieve element which is surrounded by the phloem parenchyma cells and as well as the campanule cells. Okay, so what happens, so many of the cells are transfer cells, and the transfer cells are those cells which help in rapid short distance transport. Okay, so as we can see there that sugar accumulates in a sieve element. Okay, so what will happen, that protoplasm will become more concentrated, and once the protoplasm will become concentrated, then the H2O will start flowing, okay, just to balance the concentration over there. Okay, so once the sugar comes in, then some sort of pressure is going to develop inside the phloem cells. Okay, so this pressure is actually utilized for the flow of the entire solute, right? Now have a look at this source once again. So what happens, the pressure starts to build this protoplasm, okay, and it is squeezed through the sieve pores, because we know very well that here that sip pores are there. So through that sip pores, what happens, that the sap start flowing. Okay. So what happens, due to this massive pressure, we can assume, we can understand that how big it is, because this pressure is equivalent to as high as 2.4 millipascals. Okay. And just a comparative account, if you talk about the blood pressure, that is just 0.016. So you can understand how much pressure it develops. Okay. So out of that pressure, what happens, that entire the solute start transporting from the sieve elements to the sink area. Okay. So now this entire things will go to the sink. Okay. So the actual amount of sugar and other nutrients that is transported by phloem per hour is actually called as a mass transfer, right? Now have a look at the sink. Now what is going to happen in the sink? What happens in the sink? First of all, in the phloem, sucrose is coming. So in the sink, what happens? Sucrose is actively transported out of sieve elements, as you can see there. So here is actually sucrose, right? And sucrose is actively transported back to the area, right? So what will happen? That loss of sugar cause phloem sop to be more dilute. So once it will be more dilute, then osmotic potential and water potential tend to become less negative. So as a result, what will happen? Again, the water will diffuse out. Water will again diffuse out, because here there is a more concentration and here it is a less concentration. So that water will diffuse out. Okay. So storage cells, whatever the sugar that comes sucrose, they do not store it in the sucrose, rather they store in the form of starch. So just see that what is the advantage of being storing the things in the form of starch. You see, once it is stored in starch, what will happen? That every time there is a constant osmotic pressure that is going to be maintained within this cell. Okay, because you see, every time sucrose is coming over there. So unless until this sucrose is converted into starch, the potential will keep on increasing. So that is not going to help. So some sort of trapping mechanism is required, that a constant sucrose is maintained over there, so that the water should keep coming over there to the requisite level only. So whatever sucrose is actually coming, they are eventually getting converted into starch within the sink, right? Now let's talk about one of the most important protein, what we call P protein in translocation. Okay, because you see, as phloem is under pressure, right? So what happens? That there is every chance of excessive bleeding, right? Because you see, you might have seen that you cut the phloem, then the phloem sap keep on moving out. Okay, so in order to check those laws, then there is a substances called P protein. So what P protein does? The P protein is a too large mass, so they move to the cut area, right? And they form a mesh like a structure in which rest of the thing get entangled, and finally the bleeding is going to be stopped. So this is how the P protein acts. Okay, so we popularly call them as a P protein plug. Okay, so P protein is present in all dicots, okay, but not all monocots, and in case of conifers, the P protein is absent, right? But in those plants in which P protein is not there, we have another substances what we call callose. Okay, so what callose does? Callose stays in a solution only if it is under pressure, right? The moment pressure decreases, because you see, when the cut is going to happen, the pressure will decrease, right? So callose precipitate, okay, and along the mass it goes to the area where the bleeding is going to happen. So along with the P protein or without the P protein, right, they cover the entire area, okay, and we can say that a callose plug is formed and the leaking is prevented. Right now we have seen the role of P protein and callose, which help in checking the leak of phloem in case of any injuries, right? So this is all as far as entire translocation of solute is concerned. Thank you.